So, as you may have noticed, it's possible for a T cell to receive multiple signals at the same time, and then they must decide which subset they should become. So how did they decide? Well, this is actually determined by which signals are more dominant in this process of induction. So you can see here that activated Th2 cells, Treg cells can suppress the differentiation and proliferation of Th1 and Th2 cells. So even if T cells are receiving their appropriate differentiating signals, if they're also seeing TGF-beta, they will not differentiate or become the helper T cells that they are supposed to become with the cytokines that are present. The same can be said for Th2 cells and Th1 cells and their effect on other subsets of helper T cells. So when the activated Th2 cell secretes IL-4, IL-4 will prevent the differentiation of Th1 cells. So if a T cell is seeing interferon gamma and IL-12, but a Th2 cell that has already been activated next to it is secreting IL-4, the Th1 cell will not be induced. This is very important because it prevents the mounting of both a type 1 and a type 2 immune response at the same time. It also makes sure that the body activates the appropriate immune response and doesn't toggle back and forth between the two. You'll remember from the previous infection with the leprosy patients that mounting a type 1 and a type 2 response can be essential for how you respond to an infection. You can also see that when a Th1 cell has been induced and secretes interferon gamma will in turn suppress the differentiation of Th2 cells. So even when IL-4 is present, if interferon gamma is already there because of a previously Th1-induced cell, the Th2 cell will not form. And finally, IL-4 or interferon gamma can also prevent the development of Th17 cells. So even if the cytokines that activate Th17 are present, if a Th1 or a Th2 cell has already been induced, the Th17 cell cannot form. This is essential for the body to commit to one type of response because, as we've learned, the type 1, the type 2, and the type 3 responses are very different. So as I briefly mentioned before, once these cells have been induced, they'll secrete a different set of cytokines that will then allow it to mediate its effector function. Let's review some of those cytokines, and I'll point out the ones that are important for you to remember as you move forward in this course. So Th1 cells actually secrete a lot of macrophage-activating effector molecules. These are all different types of cytokines or ligands. This includes interferon gamma, which we talked about earlier, GM-CSF, TNF-alpha, CD40 ligand, and Fas ligand. GM-CSF is important for the activation and proliferation of granulocytes. TNF-alpha is another inflammatory cytokine similar to interferon gamma. CD40 ligand, as you may remember from earlier, helps activate lymphocytes. And Fas ligand is involved in the apoptotic response of macrophages. Others that they secrete include IL-3, lymphotoxin alpha, and CXCL2, which is a chemokine that we'll talk a little bit more about later. While Th1 cells do all of these things, in the field, we typically identify Th1 cells by their ability to secrete interferon gamma. Th2 cells secrete many cytokines that are associated with barrier immunity and activating effector molecules. These include IL-4, IL-5, IL-13. Remember, these are the ones that are also important in responding to allergy. We have CD40 ligand, which again is another activating co-stimulatory molecule. And then additionally, the Th2 can also secrete IL-3, GM-CSF, IL-10, which is anti-inflammatory, TGF-beta, CCL11, and CCL17, both of which are chemokines. Again, even though all of these cytokines are secreted by the Th2 cell, in the field, we typically identify Th2 cells by their secretion of IL-4, IL-5, and IL-13. Th17 cells also secrete some barrier activating molecules as well as neutrophil recruitment molecules. These include IL-17A, IL-17F, IL-22, CD40 ligand, IL-3, TNF-alpha, and CCL20, a chemokine. We define Th17 cells in the field as being capable of secreting IL-17 and IL-22. Finally, T regulatory cells can secrete IL-10 and TGF-beta, which we know as canonical anti-inflammatory cytokines, as well as IL-35. You'll remember from the previous slides that IL-10 and TGF-beta are how we define T regulatory cells and their function. So how do these effectors actually work? Well, different types of immune responses like the type 1, type 2, and type 3 responses are highly regulated, and it's the role of these cytokines that allow them to be highly regulated. They recruit specific cells, allow for specific things to happen at the site of infection, and ensure that the patient is responding in the most optimal way to what they've been infected with. You can see here that in a type 1 response, interferon gamma is important for activating macrophages that will kill the bacteria. In a type 2 response, GATA3 expressing Th2 cells will release their canonical cytokines 4, 5, and 13 to activate specific myeloid cells like eosinophils, mast cells, and basophils. And in a type 3 response, ROR gamma T and its secretion of IL-17 and IL-22 will recruit neutrophils, which are highly important for mediating the type 3 response. So you can see, without these different cytokines, we would not be able to appropriately tailor our immune response to effectively eliminate the pathogen. Together, we'll go through each one of these different types of responses so we can understand why they're different and why they are best for responding to specific types of infections. First, we'll start with the Th1 mediated response, or type 1 inflammation. This, as you'll remember, is mediated by the Th1 cell and the general activation of macrophages to kill the infected bacteria. There are many different effector functions for Th1 cells in mediating the type 1 inflammatory response. Generally, what you need to remember is that they're important for activating the macrophages, but together we'll go through each one of their singular effector functions so we can really begin to grasp what the type 1 immune response is all about. So as we know, the production of interferon gamma and CD40 ligand on the surface of the Th1 cell will induce and activate what we sometimes refer to as M1 macrophages. These are macrophages that drive inflammation and work to destroy bacteria. Together, these cytokines will enhance macrophage killing of engulfed or phagocytosed bacteria. Another function of Th1 cells in the type 1 immune response is the induction of Fas ligand and lymphotoxin beta that's produced by these helper T cells that will then induce the apoptosis of bacteria-laden macrophages. That means macrophages that are occupied by bacteria will die, not making sure that the bacteria cannot continue to replicate and infect the patient. It kills chronically infected cells, and it releases the bacteria to be destroyed by macrophages that are still fresh and capable of killing the bacteria. Another effector function is the production of IL-2, which you may remember is highly important for inducing the proliferation of CD4 positive T cells and CD8 positive T cells. Together, this can alter the balance of a Th1 versus TFH differentiation to favor the Th1 induction or the type 1 immune response and supports the expansion of CD8 T cells. You may remember that T follicular helper cells are important for activating B cells. However, when a person is infected with an intracellular bacteria, the appropriate response is a CTL or CD8 positive cytotoxic T cell response. This is because MHC class 1 loads intracellular bacterial peptides onto the surface of the antigen-presenting cell, and that a B cell is typically better at responding to antigens that are expressed on the outside. All of this is here now. 3 more functions, which I'll go through one by one. First, they produce IL-1 and GM-CSF. This stimulates the production of monocytes in the bone marrow. Monocytes are precursors for macrophages, which, as you may remember from the first panel, are important for killing the bacteria that's infecting the host. This allows for a higher number of macrophages to go into the site of infection and help with clearing of the bacteria. Th1 cells also produce TNF-alpha and lymphotoxin alpha, which act on local blood vessels. This allows the endothelium to induce macrophage binding and exit from the blood vessel, so when the macrophages or monocytes are released from the bone marrow, they can actually traffic into the site of infection by leaving the bloodstream and going into the infected tissue. And finally, Th1 cells induce CCL2. This is a chemoattractant, or a chemokine, that draws monocytes towards the site of infection, where they can differentiate into macrophages and help with bacteria killing. So the main takeaways from the Th1 mediated response, or the type 1 immune response, are that macrophages must be activated and recruited to the site of infection. This allows for increased phagocytosis of the bacteria at hand, increased killing of the bacteria, and faster resolution of the infection. Th1 cells also help activate cytotoxic CD8 T cells and suppress the B cell response, such that infected cells can be directly targeted and killed. Th1 responses in this scenario are augmented, while Th2 responses are suppressed or shut down. A chronic Th1 response can also result in the formation of a granuloma. This is when T cells and other myeloid cells surround the infection and eat it away little by little. This is often a long-term effect of being infected and is not often resolved in a short period of time. The next immune response we'll talk about is the Th2 mediated response, or the type 2 immune response. This is characterized by the production of IL-4, IL-5, and IL-13 from Th2 cells, which we'll see act on the bone marrow and goblet cells in the epithelial layer. So Th2 cells produce IL-13, which induce epithelial cell repair and mucus. This increases cell turnover and helps with the shedding of the parasite or the helminth from the host. Parasites and helminths are very large and can often not just be eaten by macrophages. Instead, they must be expelled from the host. This increased turnover aids in the expulsion of the helminth or the parasite, since it can't be directly targeted by myeloid cells like macrophages. IL-13 also increases smooth muscle contractility and enhances the worm expulsion. So the more muscle contraction you have, the faster you're going to push out this helminth or parasite. Th2 cells also recruit macrophages of the M2 subset, which means that they're better at responding to a helminth or parasite infection as opposed to an intracellular bacteria like the M1 macrophages. This increases the production of arginase 1 that increases smooth muscle contraction and enhances the tissue remodeling and repair we talked about in the first 2 panels. IL-5, produced by the Th2 cell, recruits and it activates eosinophils. Eosinophils are important myeloid cells that are needed to respond to helminth and parasite infection. They produce MBP, which kills the parasites, and they can also mediate antigen-dependent cytotoxic killing using parasite-specific IG. Th2 cells, finally, will also drive mast cell recruitment via IL-3, IL-9, and specific IgE. Mast cells will then produce these effector molecules like histamine, TNF-alpha, and MMCP. These further recruit more inflammatory cells to the site of infection to enhance the things that we talked about in the first two panels, like cell turnover and increased expulsion of the worm, helminth, parasite, etc. So the main takeaways from the Th2 mediated response, or the type 2 immune response, are that Th2 cells activate epithelial cells, eosinophils, mast cells, and basophils to fight against an invading parasite or helminth. They increase the production of mucus, which then helps the expulsion of the pathogen, as well as increase muscle contraction. And finally, they encourage the production of IgE and histamine that target the invading pathogen and aid in its expulsion from the host. Finally, we have Th17 mediated responses, or the type 3 response. In this type of immune response, Th17 cells will produce IL-17 and IL-22 that induce the production of antimicrobial peptides from epithelial cells. These are nonspecific peptides that will target foreign pathogens and cause them to lyse. Oh, what's happening again? The IL-22 produced by Th17 cells will also increase epithelial cell turnover. This prevents the extracellular bacteria from grabbing onto the epithelium and working its way into the body, instead resulting in its expulsion from the host, similar to the parasites and helminths we just talked about. IL-17 will activate stromal cells and myeloid cells to produce GCSF. This stimulates a neutrophil production in the bone marrow. As you might remember from previous slides, the type 3 response is highly neutrophilic, meaning that there's a lot of neutrophils at the site of infection, and they carry a lot of short-lived innate effectors that will aid in killing the bacteria. IL-17 will also activate stromal and epithelial cells to produce the chemokines that recruit the neutrophils being produced in the bone marrow, and produce CCL20, that's a chemoattractant for other Th17 cells. So it essentially amplifies the response and helps all of these other panels occur. So the takeaway from the Th17 mediated response, or the type 3 immune response, is that Th17 cells induce mechanisms that recruit massive numbers of neutrophils, which produce short-lived innate effectors that kill the extracellular bacteria. Th17 also drives the production of antimicrobial peptides from the epithelium to encourage the expulsion and killing of the extracellular bacteria. And finally, increased proliferation and sloughing off of epithelial cells aids in the shedding of the bacteria before it can infect the rest of the body. So we've talked extensively today about CD4 helper T cells, but what about the CD8 positive cytotoxic T cells? Just because there aren't a multitude of subsets doesn't mean that they aren't important. This brings us to our last objective for today, which is to discuss how cytotoxic T cells kill a target cell. So, most of the effectors produced by CD8 positive T cells are designed to kill infected cells. These include effector molecules like perforin, granzymes, granulysin, and Fas ligand, which we'll talk about, as well as interferon gamma, lymphotoxin alpha, and TNF-alpha. So before the cytotoxic T cell can kill the infected cell, it first has to make contact with the cell. It'll see whether it's carrying the antigen that it recognizes. If it doesn't, then the T cell will move on and look for another infected cell. However, if it does, it will prolong its contact with that infected cell and release those cytotoxic molecules that we talked about, leading to the apoptosis of the infected cell. This contact that's formed by the cytotoxic T cell and the infected cell is called a supramolecular activation complex, or a SMAC. These SMACs can also be called immunological synapses and are more than just a point of contact between the two cells. The SMAC is composed of an outer peripheral zone called the P-SMAC and an inner central zone called the C-SMAC. You can see that here on this diagram. Once the P-SMAC is formed, TCR molecules that initiated the contact between the infected cell and the cytotoxic T cell will then migrate to the peripheral SMAC. This creates a nice open space where cytotoxic effectors can be released onto the infected cell. Another thing that happens once the contact is initiated is the TCR will also be endocytosed into the cytotoxic T cell, so it can't get in the way of the contact between the killer cell and the infected cell. This creates even more space for those cytotoxic effector molecules to be released. This is important because it prevents the killing of neighboring cells and only kills the directed cell via apoptosis. So you can see here, the CTL is recognizing and binding a virally infected cell. It creates a small specific place for the release of cytotoxic molecules like the granzymes we talked about. It'll then program that infected cell for cell death by inducing DNA fragmentation. Once that cell is ready for cell death, the cytotoxic lymphocyte will move on and find another infected cell, while the first target cell is dying via apoptosis. So how does the CTL actually induce apoptosis? We're going to walk through that together on the next slide. So apoptosis is primarily mediated by the location of cytochrome C within the cell. In a normal, uninfected cell, cytochrome C, which is a component of the mitochondria, is present only in the mitochondria. However, when cell death is induced, the mitochondria will begin to swell and leak. Cytochrome C is then no longer able to stay within the mitochondrial matrix and is released into the cytosol. There, it can bind and induce a conformational change in a molecule known as APAF1. Together, APAF1 and cytochrome C will assemble into a complex called the apoptosome, which recruits and activates multiple copies of another enzyme called procaspase 9. When procaspase 9 becomes assembled, it then cleaves and activates procaspase 3 into caspase 3. Once caspase 3 has been activated, it cleaves ICAD, releasing CAD to enter the nucleus and cleave DNA. This then drives the death of that targeted cell. So how does this happen? Well, the effector molecules that are released by the CD8 positive T cell will actually make sure that this apoptotic program is induced in the infected cell. Perforin, which is released by the CTL, will aid in delivering contents of granules, so granules are these little pockets that contain the other effector molecules, into the cytoplasm of the target cell. Granzymes are actually serine proteases, and these activate apoptosis in the cytoplasm of the target cell. Finally, granulysin has antimicrobial actions that will further induce apoptosis in the targeted cell. So let's walk through this together. First, engagement by the TCR to the peptide MHC complex on the infected cell will cause directed release of perforin in that space created by the C-SMAC, as well as granzymes that will then go into the cytotoxic T cell, the virus-infected cell. Granzyme B is delivered into the cytosol and the infected cell via pores formed by perforin. So perforin will first line the surface of the infected cell such that granzyme can go through the little pocket formed by perforin and begin mediating its effector function. Here, granzyme B will target BID, another apoptotic protein, and procaspase 3, which you'll recall is the enzyme responsible for ultimately causing the fragmentation of DNA and thus its death. BID, or truncated BID, which is cleaved by granzymes, will disrupt the mitochondrial outer membrane. This, of course, causes cytochrome C, that very important mitochondrial protein, to be released into the cytosol, where it will activate caspase 3 and cleave ICAD via that mechanism we talked about earlier. And then CAD, the DNase, that is a product of ICAD cleavage, will go into the nucleus and begin to cleave DNA, resulting in the death of the targeted cell. Release of cytochrome C into the cytosol activates further apoptosis, eventually allowing the cell to apoptose and the CTL to move on, knowing that it's done its job. Remember that I said earlier that the way that a cell is killed via the formation of the SMAC is important for making sure that neighboring cells are not affected by the effector molecules released by the CD8 T cells. You can see here that after the CTL recognizes an infected cell, it can very directly kill the targeted cell and leave the neighboring cells intact. This reduces the amount of damage that's created during the immune response. So before we finish today, let's first go over some of the objectives we talked about earlier so you can get a sense of what you should have taken away from today's lecture. First, we worked to understand the two major subsets of T cells, the CD8 positive cytotoxic T cells and the CD4 positive helper cells. We remember that cytotoxic T cells, the CD8 expressing ones and the CD4 expressing helper T cells are determined early on in development in the thymus. We also learned that CD4 positive helper T cells have a multitude of roles, including further activating CD8 positive T cells, B cells, and aiding in the recruitment of other innate immune cells. And we also learned that CD8 positive cytotoxic T cells can directly kill infected cells. Secondly, we learned the roles of the major CD4 helper T cell subsets and how they're induced. I'm not going to go through this table again now, but this is something that you can go back on and look at when you're reviewing for your exams. The third objective for today was to know the effector function of helper T cells. We learned that Th1 cells are important for the type 1 inflammation, where macrophages will be activated to kill bacteria. We learned that Th2 cells are important for responding to helminths and parasites. We learned that the Th17 T cells are important for responding to extracellular bacteria. And we know that there are two special subsets of helper T cells, T follicular helper cells, which are needed for B cell activation and maturation, and T regulatory cells, which are important for causing the contraction of the immune response once the infection has been cleared. And finally, our last objective was to discuss how a cytotoxic T cell kills a target cell. We learned that in order for this to happen, the cytotoxic T cell must first make a very complex interaction with the infected cell via a complex called the SMAC. We also learned that the formation of a stable P-SMAC or peripheral SMAC allows for the opening of a space in the center of that interaction where cytotoxic granules can be released to directly kill the targeted cell. We learned a little bit about some of these cytotoxic granules, including perforin, granzyme B, and others. And we finally learned that these cytotoxic molecules will induce apoptosis via cytochrome C release in the infected cell, allowing the infected cell to die without affecting the neighbor cells, minimizing the amount of damage that occurs when a cell is infected.